Hello, welcome to Bio 1006 Biochemistry and Cell Biology. For this, I'm going to be doing a lecture summary recording about the first lecture we've had with Dr. Rushworth about lipids, fats and fatty acids. This PowerPoint is going to be very interactive in the terms of meaning that I'm going to be asking questions throughout this PowerPoint, and when I do so, I will also ask you to pause the video. Okay, when I ask you to pause the video, just pause it and answer the question that's on the screen. Then once you've answered the question, resume the video, then we'll compare and contrast the answers we've got. So without further ado, let's get going. Okay, so the learning objectives from that lecture was to know the definitions of lipids, fats and fatty acids, to be able to list several biological functions of fatty acids, to recall several diseases that are linked to obesity, to understand that triglycerols, also known as triacylglycerols or fats, are made up of a glycerol backbone esterified with 3 fatty acids, to understand and know the definitions of saturated and unsaturated in terms of fatty acids, to understand what cis and trans isomerism means in terms of the structure and melting point of fatty acids, to be able to recognize and to draw triglycerides and fatty acids, and finally to be able to understand stick diagrams of biological molecules. So what diseases or disorders can be caused by an unhealthy diet? Okay, so from here I want you to make a mind map about the diseases and disorders linked to an unhealthy diet. So pause here and resume once you've done. Okay, so let's see what answers you got. Did you get CHD, type 2 diabetes, osteoarthritis, hypertension, anemia, malnutrition, infertility, organ failure, or even at least cancer? If you've got at least some of these, give yourself a tick. If you've got some extras which I have not listed, well done to you. Talk a little about adipose tissue. So fat is stored in specialized cells called adipocytes, which have the ability to store a very large amount of fat. The nucleus gets pushed to the size of the cell, allowing more space for the fat to be stored. So on this diagram here, you can see that this white area here, this is just pure fat. And these areas here, which you can see the nucleuses, and the little black dots are the nucleolus. So as you can see, they're all pushed to the far extremities of every cell to make room for all this vast amount of fat that's been built up inside. Lipids. So lipid is a molecule which consists of, pause here. So a lipid is a molecule which consists of a glycerol head and fatty acid tails. If you've got both of those, give yourself a tick. If you've got anything more, that's probably not a lipid. If you've got anything less, you definitely need to revise the topic of lipids, because these are the two main components of a lipid. So again, this is another type of lipid. It's called a phospholipid. So again, it's still classed as a lipid. The only difference is on the glycerol head, it's got a phosphate group attached. Okay, and this makes the phospholipid polar. Okay, so we now have a hydrophilic head and a hydrophobic tail. So the hydrophobic tails will point away from water, and the hydrophilic head will point towards water. So now I'm going to talk a bit about fatty acids. Okay, so there are two main types of fatty acids. We've got cis and trans fatty acids. Okay, a trans fatty acid. So here we've got the carboxylic acid terminus end here. Then we've got a chain of the fatty acid. Then we reach to a carbon-carbon double bond. Okay, for a trans fatty acid, one hydrogen will be on one side, the other hydrogen will be on the other. Okay, what this will do, if we then look to the diagram on the right, this will cause the molecule to be in a straight chain like this. Okay, if we compare that to a cis fatty acid, again we've got the carboxylic acid terminus, follow along the fatty acid chain until we reach the carbon-to-carbon double bond. Both hydrogens are on the same side of the double bond. And what this does, it induces a kink in the fatty acid. So rather than have this long straight chain like the trans form, we then have this L-shaped kink in it. Okay, so the definition of a fatty acid is that a fatty acid is a long hydrocarbon molecule with a terminal carboxylic acid group. Okay, so we just go to the previous slide, we can see here, from here towards the end, this terminus point here, this is the carboxylic acid group. Okay, and you just got to make sure you remember this. Okay, fatty acids are a very basic component of biological life, so you have to make sure you remember the structure of a fatty acid. Okay, now we're going to discuss the difference between a saturated and a non-saturated fatty acid. Okay, so for the cis and trans fatty acids, these only apply to non-saturated fatty acids. The reason being that non-saturated fatty acids are the only type that have this double bond in it. If we compare that to a saturated fatty acid, we can see that all the hydrogens are attached to a carbon, so there's no carbon-to-carbon double bonds. Okay, if you think about it like this, Imagine you're out in the rain and you're wearing like your favourite jumper. Okay, obviously you wouldn't actually wear your favourite jumper in the rain, you probably put a coat on. But when you get covered in water, you tend to say that the jumper is saturated with water, meaning that it's completely surrounded by water. But in terms of fatty acids, it's the carbons being completely surrounded by hydrogens, not having any sort of gaps like this. Okay, so saturated fatty acids, so these tend to have a higher melting point than the non-saturated. This is because if you look at these compared to these, if we go back to the previous slide, okay, these fatty acids, imagine we've got these kinks and stuff in these. This will make these very hard to form long straight chains. So as a result, the saturated fatty acids can stack on top of each other very easy, whereas in comparison to a non-saturated, they will be all over the place. There'll be complete disorder amongst them. So as a result, because they will be all nice straight chains, there'll be strong intermolecular forces between them. As a result, saturated fats will have a higher melting point, and more than likely, they will more tend to be solid. So for example, butter is a very good example of a saturated fat. So now I want to label the several different functions of fatty acids. So pause here. Okay, so the functions of fatty acids are to act as an energy source, for protein modification, to act as hormones or protein signaling, or components of the cell membrane. Okay, again, similar to the definition of a fatty acid we've had, you have to make sure you remember these. Okay, just by chance, what if there's a question on the use and functions of fatty acids? Okay, so it might be worth considering to remember these 4 functions of a fatty acid. Okay, so now let's quickly give yourself a test. Okay, so I'm going to ask 3 questions, and these are all going to be quick fire questions, okay? So, you have a choice of A, B, or C. Okay, so question 1, which of these is a saturated fatty acid? Is it A, B, or C? The answer, A, because it has no carbon-to-carbon double bonds and it's completely surrounded by hydrogen. Okay, question 2, which of these, A, B, or C, is a cis fatty acid? The answer is B, because it's got both of the hydrogens on the same side of the carbon-to-carbon double bond. And question 3, which of these 3 will have a higher melting point, A, B, or C? The answer is A, because these can stack easily on each other, get packed nice and tightly together, and have strong intermolecular forces between each other. So if you've got 3 out of 3, well done to you. If you've got 2 out of 3, you might need to check up on the answers you're not too sure on. If you've got 1 out of 3, you might need to start considering revising. And if you've got 0 out of 3, you've definitely got to start revising this, because 0 out of 3 is quite a low mark to get. And finally, I'm going to talk a little bit about esterification. Okay, so esterification is a process of when an alcohol group and an acid group fuse together to form an ester. Okay, so here we've got glycerol, or the glycerol head, and a fatty acid, where R on either of these molecules means any number of hydrocarbon. It could be a methyl group attached, or it could be as long as 10, 20, 30 different carbon groups. Okay, so looking at this diagram, what do you think happens between glycerol and fatty acid in order to produce our triacylglycerol? So pause here now. Okay, so let's have a look. So let's just take into consideration what we've got here, because this is the part that's going to be reacting with each other. Okay, so this carbon will be bonded to an oxygen, and that oxygen's bonded to a carbon. So what's going to happen here? Let's just use this middle one as an example. So we've got an OH and an HO, and we know that there's going to be an O remaining. So let's just have a look here. Oh, we've got a H, a H, and an O. What could possibly happen between them? So these will bond together, giving us the lipid. And this gets released, and look, we have water. So it's a condensation reaction, meaning that water is released. Okay, just to finish off, I'm going to set you 3 exam style questions just to have a crack at. But what I want you to do, I want you to make sure that you make your own mark scheme for each of these questions, okay? I've set out how many marks it's worth, but what I want you to do, I want you to try and set out a mark scheme. So what do you think I'll be looking for in the answers for each of these questions? Okay, so question 1, outline the differences between a cis fatty acid and a trans fatty acid. How does the structure of a lipid allow cells to be kept in membrane bound? And then C, define what is a fatty acid and a lipid, and draw diagrams to illustrate their structures. So thank you for watching. I'll probably make another one of these depending on what feedback I get, and I hope you guys enjoyed. Peace out.